Hi, my name is Blaze. Welcome to my genetic regulatory network playlist. In this video, I will cover one of the introductory concepts of controlling translation post-translational modification. So the playlist goal is to show various Python modeling techniques applied to biological system. Not there yet. And here I will provide a conceptual understanding of how to control gene expression via translation and post-translational modification. If we go forward, or if we think about the central dogma of molecular biology, translation is the act of turning mRNA into protein. And the post-translational modification is turning this peptide amino acid sequence into an actual functional active protein. And so we can insert ourselves in these processes in various ways to have control over the output of our active protein. I wanted to start off with the basic understanding of the prokaryotic gene structure, and I wanted to simplify translation. First of all, this is our DNA, where we have the promoter region like we talked about in the last video. And within this DNA, there's going to be a ribosome binding site. The ribosome's not going to bind there. It's not RNA yet, and it's not in a place where a ribosome can get to. So this doesn't do much when it's on our DNA structure. Then we have our actual coding sequence. This sequence will actually end up being our protein output. And we also have a terminator both for the ribosome, and there'll be some termination further downstream where the RNA polymerase holoenzyme will dissociate, as I talked about in the last video. Now, when going from mRNA to protein, we have, based on our transcription start site, we have all of this region here based on to where our transcription ended. But within it, we have a ribosome binding site. And this is important because the ribosome is going to be pretty fundamental to translation, and we'll start translating at our AUG start sequence. That adds a specific amino acid and starts the sequence of recruiting transfer RNAs to add amino acids onto our growing peptide chain. This will occur throughout this coding sequence until you hit a stop codon listed here. The output is a peptide chain of this sort. Now, this isn't an actual functioning protein. It may need some post-translational modification, and I'll talk about that in a second. Note that this is a prokaryotic gene structure. A eukaryotic gene structure would have introns and extrons, and those would have to be spliced out in some way, along with kind of more complications on where where ribosomes are located. But since in E. coli and in most prokaryotes, there isn't a nucleus, so we won't have to worry about that here. So to continue our discussion on how we can control translation and post-translational modifications, I made this figure just using Inkscape. Maybe I'll do a tutorial on that someday. Inkscape is a very, very useful open source design software that you can use to make basic images. Anyway, essentially, right, we have our transcriptional control, which I talked about in the last video. Then when we get on our mRNA, it gets a little bit more complicated. There's a lot that happens with mRNA. First of all, we have our ribosome binding site, so we can activate or repress this binding site depending on our design. Now, this can be done by the mRNA actually having some cleaving ability of itself, so that's why I have this arrow here. So RNA is thought to be one of the first kind of enzymatic things that existed on Earth, and those are what we call ribozymes. They have enzyme-like activity, and they can cleave stuff. They can cleave themselves. So that can play a role if you design a ribozyme to cleave out the RBS. This mRNA also has kind of a degradation constant, depending on the activity of other degradation proteins, or whether this mRNA sequence gets recruited to certain lysosomes, which are the garbage disposals of the cell. That rate will be higher or lower, so we can control that. But just to show you kind of how long an mRNA might last within a cell, bacterium half-life is in minutes, And so this looks like it drops off after about 4 minutes. In yeast, drops off maybe about after 20 minutes. And then in humans, mRNA can last, it drops off after about 10 minutes. So the idea of this being like different organisms have different degradation constants, you might want to control this degradation constant a little bit better if you're just working with bacterium. Then you go into translation, so the actual act of the ribosome translating, and you get this kind of unfolded protein. The modifications you can control, so if you get some kind of truncated protein that's not going to fold correctly, that's just going to get degraded and have no function, which again, we can control the degradation function similarly to as we control the mRNA function. But with the modifications, right, if we took away the modification protein, then this protein's not going to be modified, it's not going to have that cellular function, and we're going to lose that function. And so you can control gene expression in that way. Something else to understand is with this ribozyme, we can actually make this ribozyme cleave itself before, say, the protein would need to in order to become a fully functional protein. What this would do is result in a short-chained protein that would end up not having any functionality because it doesn't have its full chain length and can't fold properly and thus can't have its structure that it would like to, and it can't interact with other molecules in the way that it would like to. That brings up the idea of protein primary, secondary, tertiary, and quaternary structure. So the primary structure of a protein is just the peptide sequence, just the amino acid sequence. Then the secondary would be any folding, first level folding of this sequence, so like beta sheets or alpha sheets. And then secondary would be bigger folds than that. Third would be bigger, even bigger folds than that. And quaternary would be its interaction with other proteins in order to actually become fully functional, a fully functioning unit. So you can control any of those because oftentimes those require external proteins to fold and process and control that modification. Anyway, that's about it for the big picture of controlling translation. And so in the next couple of videos, I hope to show you how we can model that based on what we know and reaction times of what we know of these types of modifications.